So I'm going to be doing a couple of things today. I want to make sure we're kind of on the same page of what is the value of genetics and why would we even bother doing a genetic test. And then I'm going to touch on a little bit about, for those that maybe aren't so familiar with 3x4, is what makes us different? Why are we a little bit different from other genetic testing companies you may have used or heard about? And lastly, I am going to give you a demonstration of what we call our interactive blueprint, which we recently launched with Rupa. So we got a lot to get through. So I am going to jump in. Please put comments in the chat, but also I will stay as long as I need to answer any questions that you have. Right, let's jump in. So we're at an interesting time in genetics or genomics, where we're hearing a lot about both genetics, but also about epigenetics. And there is this kind of sense that we need to choose. We just say, well, genetics is more valuable, but I'm just going to do that. Or why do genetics when I can just do epigenetics, which is kind of what is happening. So this is my vision of genetics and epigenetics. It's a yin and yang relationship. And as we go through my slides and the demo of the interactive blueprint, I'm going to keep on coming back to this idea that we want to always think about genetics and epigenetics. And I'm going to show you why. Okay, so we're going to start off just a few slides. What is genetics? What is epigenetics? So when I talk about genetics, I'm always talking about the DNA sequence code. It's got 4 letters, A, T, C, G. And I realize some of you listening in are probably experts about this. So just bear with me. 4 letters that determines our whole code. And when we group these letters together, we make something called a codon. And a codon really is just a code for an amino acid. So you can see in the slide, we've got all the letters, and each 3 is making an amino acid. When we put our amino acids together, we make proteins. And of course, our proteins can be anything, our enzymes, our hormones, neurotransmitters, and that makes our body work. So when we have genetic variation, remember 99.9% our code is the same, 0.1% we're going to be a little bit different. That 0.1% is what we're so excited about. It's what we're interested in. How do those 0.1%, I call them spelling changes. That's all they are. They're not mistakes. They're not mutations. They're not defects. They're just spelling changes. I might have an A, you might have it. How does that make us different? So we talk about genetic variation as giving us insight. And when you do a 3x4 genetic test, that's what we're testing. We're testing to see those spelling changes. Which version have you have? Not good, bad, right, or wrong. Which version does you have, and what insight does that give me? Okay. And this is what it looks like for those of you that haven't seen it. You will get the name of the gene. We always take a very complicated long name and put it into 3 or 4 capital letters. Then in the middle, in the gray, we have the SNP name, the gene variant name. Tells us whether some people have a T, some have a C. And then the right-hand column, we're seeing the actual genetic result of the patient. So now, that was like genetics 101 in 1 minute. Now we're going to do epigenetics 101 in 2 minutes. So when I talk about genetic variation, I talk about insight. When I talk about epigenetics, I say epigenetics determines action. And by action, it means what are the diet, lifestyle, supplement, testing, kind of environmental choices that your patient's going to make, and you're going to guide your patient. So why do I say that? So epigenetics, epi is above. So epigenetics, it's above genetics. It means anything to do with genomics that isn't about that sequence code, the A's and T's and C's and G's. So epigenetic changes are modifications to our DNA that regulate whether our genes are switched on and off. We use the terminology of switched on and off. We sometimes use the terminology of the gene is expressing or it's expressed itself, or the gene is silenced. And both of those are very valid. And these little modifications are attached to our DNA, but they do not change the sequence of that DNA. Okay, I love this slide. It's probably my favorite slide of all. So when I started in nutrigenomics, which was 25 years ago, I was a dietitian before I went into genetics. And I only thought about how the food we eat, the exercise we do, and maybe I had a concept around toxins in our food and how that would change how our DNA switches on and switches off and expresses itself. And remember, when a gene expresses and switches on, it makes a protein, again, enzyme, hormone, neurotransmitter. When it switches off, it stops making the protein and you don't have that enzyme, you don't have that hormone happening. Okay, So this slide was like a huge aha for me of saying what actually will impact my gene expression, my switching on and switching off. And as you can see, every single thing we do every minute of the day is going to change the way our genes behave or express themselves. And, if you start on the left-hand side and we go around, there's some obvious ones. The drugs we take, pharmaceuticals, will change gene expression. In fact, that's often their job. Our mood, think about when you're joyful and happy, how that will switch on genes. But when we're depressed and sad, that's going to change gene expression. Our microbiome, of course, affects everything. Toxins, whether they're environmental toxins, toxins in our food, drug abuse, complementary medicine, love this. Reiki, kinesiology, acupuncture. Why is acupuncture so impactful? It's switching on or switching off genes. Our body weight will impact our genes. Seasons. So I live in Seattle. I always use it as a perfect example. I'm starting to go into winter and I can feel it happen to me, the gray, the dark, the rain, how it's changing some gene expression, which is changing some of my enzyme, which is changing my neurotransmitters. And that's kind of seasonal affective disorder. Obviously, all the food we eat, diseases we're exposed to, social interaction, connection, conviviality, eating with people, not eating alone, being isolated on the other side of the stream. What happened in COVID, that deep isolation and loneliness changed our gene expression. And of course, exercise and anything like gamma rays we're exposed to. So we need to think about epigenetics as every single exposure and how is it impacting our genes. Okay, so now that we've got a good sense, and epigenetics is way more complicated than that, but this is obviously not talk about epigenetics. I just want you to think about insight and action. We look at our genes to understand who we are and how we respond to the world around us, lifestyle, toxins, stress, trauma, exercise, hormones. And then we craft a diet and lifestyle supplement recommendations, which is going to impact our epigenetic expression to optimize the health. And that kind of brings that yin yang together. So now we move into my next theme, which is why bother with a genetic test? So this is actually a question that was sent to me in an email by one of our clinicians. She's given me permission to share it. How does and this is so relevant for Rupa, right? Because this is in essence what you do at Rupa. How does genetic testing change my clinical approach and treatment? It seems to me that all the systems we look at, I'm adding in here genetics, are addressed anyway in functional tests when patients present with the symptoms anyway. So why bother with a genetic test? Okay, I am going to give you my short version. Normally this is like an hour presentation. So I'm trying to give you a touch of all the things that I think about in genetics. So remember one thing, genetics is a once-off test. Your patient does it once in their lifetime. Their DNA sequence code does not change. They do not need to retest. We'll add new genes pretty much every year. We just launched a new version into the test where you will always be able to come to us and say, I want the new version of the report. Your patient doesn't swab again. Your patient doesn't pay again. Okay, putting that aside, why is genetics valuable? Remember that genetics is the source code that determines, so almost think of hardware and software. Genetics is the hardware. It's the code that is the blueprint. And then the things we do in our life will be the software that changes the expression. Okay, so here's my reasons about why genetics is so valuable. Number one, when you're working with a patient, you've got their labs, you've got their medical history, and of course you've got the symptoms and the goals that they want to achieve when they come to you as a clinician, and it's a lot of information. We want to get to the answer as quick as we can. We want to intervene. I always say 20% intervention for 80% impact. How do we figure that out without trial and error? So the first thing about genetics, it helps us know where to start. We always say and this is where the name of the company comes from, 3x4, what are the three most important areas I can focus on? Is it detox? Is it inflammation? Is it histamine? Is it mood, neurotransmitters? Know where to start. The second thing, last one, I could talk about it for hours. We don't know what we don't know. So no matter how brilliant we are as a clinician, there is always going to be a clinical gem that we're going to get in genetics that's going to add to our insight about our patient to get to that intervention answer, that treatment answer, without the let's try this. Okay. And I give a presentation where I talk about all different examples of how we don't know what we don't know. It might be about gluten. It might be about blood clotting. It might be about how we break down dopamine and serotonin, or how we don't break down dopamine and serotonin. I can go on forever on that. The third one, personalization of diet and lifestyle recommendation. That is what we're all about. That's why we're doing tests. We want to make sure that we are personalizing the treatment plan that we're giving, and we're not just giving a standard protocol. Precision testing. Obviously, this is all about RUPA. So we do a huge amount of work about connecting genetics and testing. And that could be a baseline blood test. It could be glucose, insulin, potassium, sodium, HCRP. It could be your baseline. And of course, it can be the more kind of intricate and amazing functional tests. And I think of the Dutch test. I think of organic acids. I think of micronutrient testing. So we talk about precision testing. Genetics is a screening tool that helps us figure out what other tests do we want to order for this patient. We know they're expensive. We know they take time. Sometimes they're invasive. How do we figure out the most meaningful and efficient tests that we're going to order? So we call that precision testing. And lastly, genetics helps us with targeted supplementation. How do we order the supplements that are going to have the greatest impact on that epigenetic effect, and not kind of what I always say, that kind of, I'm just going to order all these supplements as an insurance plan that some of them are going to do their job. We really want to target our supplementation. So that's where I feel the value of genetics is. And so we can talk about it more. Now we're going to shift into what makes 3x4 different. So I've been building genetic tests for 25 years, and many companies that you may have come across. But when I started 3x4, I really wanted to think about what have we been doing in genetics for so many years, and how can we do things better? There were many things in the world of genetics where we went down, in my opinion, obviously, the wrong road, the wrong journey. And I don't think we served clinicians well, and I don't think we served our patients well. So you will see in the things that we've done, we're trying to think about all the touch points that you would experience in the world of genetics that we can do better. So we're going to, the science, we're going to touch on that, the report, and there's two versions of the reports. Mentorship is a really big deal for us. Education, it's a given. And we know that's one thing we do beautifully. We've done a boot camp with Rupa. We're doing another boot camp. I think it's coming in the beginning of 25. And community. So it's important to know that genetics is not just a report that you get. It has to be all these things to help you extract the value, the clinical value. All right. So how always think, the most important question you have for your patients is, how do you build a genetic test? And any genetic company that you choose to use with, you should be asking this question, but how did you build your test? And the most important question is, how do you treat genes? How do you treat SWAP? So there's two big differentiators at 3x4. The first is the concept of a polygenic approach. So one of my, and I know I'm speaking fast, but we've got a lot to get through, so forgive me for that. Someone will tell you I always speak fast, but anyway, one of my great concerns in the industry was a single SNP. I am going to test MTHFR and I'm going to build a treatment protocol on it. I am going to look at ApoE, I'm going to look at count, and I'm going to make some assumptions about what I'm seeing. But the reality is, the SNPs that we test, which I call the diet and lifestyle genetic SNPs, are not medical genetics. They don't give you, let's say this, if you have a SNP, you're going to get this disease. They're what we call low impact or low penetrance. So by themselves, they're insightful, they're informative, they're interesting, but they're not powerful enough to create whole diet and lifestyle recommendations on one step. So I moved to a polygenic approach, which is very well validated in the literature. But the problem in the literature is that all the polygenic scores were based on disease. So I can look at a group of genes to determine my susceptibility to diabetes. It's interesting. It's not clinically valuable. So we always think in a functional integrative approach, but what's happening at a biochemical level? How do we take a validated methodology, which is polygenic, and apply it to biochemistry and physiology instead? Let's help us understand what are the impact of genes on these biochemical processes. Okay, so this is what we do. And if any of you have seen the 3x4 report, or you can always download from the website, this is one of the core pages in the PDF version of the report. So we've got two versions of our report. One is the PDF. It's very beautiful. It's got colors and stories and pictures. The pictures change. And then we have the interactive one, which I'm so excited to show you today. We're going to get there. But this is from the patient-facing one. We use color, not traffic light red. I don't want to have a red conversation. Wherever you see dark purple, the genes are having a very high impact, purple a high impact, blue a medium impact, yellow a low impact. So what you're seeing here are 36 of these pathways. A pathway is a polygenic score or total genotype score for a biochemical or metabolic area. And we divide them into 6 categories. So we always want to think upstream. We always want to think root cause. How do we get that, if you know what's happening at that cellular level, and then work down? So 6 categories. You'll see cellular will always be the first place I will look. Your, inflammation, detox, methylation, oxidative stress. Then we go down to systems level. I don't want to spend too much time on this. Cardiovascular, energy is about weight. Why do we gain weight so much? Find it hard to lose weight. Activity is about exercise and training or response to training. And then, of course, nutrients. Okay. So then the next question is, how do we choose the SNPs that we include in our report? We are probably one of the most conservative genetic companies. We don't offer 1,000 SNPs or 25,000 SNPs or 100,000 or a whole genome. There is this idea that if there are more SNPs, it's a better test, but actually the opposite is true because it's data if it doesn't have clinical value. So how do we determine that clinical value? And as I put here, not all SNPs are equal. So we built something called Darwin. It's another methodology that we have patented. It's called Darwin because it's a process to determine, say, only the fittest genes will survive and be included in our report. So what is Darwin? Darwin is based on the ACCE model. Again, another methodology we use. It's actually 4 criteria. I'm just sharing the main 2 criteria: scientific validity and clinical utility. And I'm sure you're familiar with this. Scientific validity is basically asking the question, how good is the science? Clinical utility is, how clinically useful will this be for me? So this I've already showed you. It's the blueprint report. I'm going to move on from that slide. So when we look, I'm actually going to go back to this. There are 8 criteria we evaluate. The first 4 are the scientific validity. I thought I had another slide on this. I took it out, so I'm going to go back. 4, basically asking, how good is the science? How many articles? Are they replicated data? Did they get the same results in vivo, in vitro? All the things you would normally look at to determine good science. It's the clinical utility that is really hard to evaluate. It's also the clinical utility that is the most important to know. So the criteria for that is, does the SNP change biochemistry or physiology in some way? If it's an association, if it's a GWAS study, we're not interested. We want to know there's a change. The second thing is it associated with a phenotype? So is the SNP associated? It could be blood glucose or insulin. It could be liver metabolism or inflammation. It could be hormone changes, but there needs to be a phenotype. The third thing is there a biomarker? And this is, of course, where Rupa comes in and the concept of precision testing. Is there something I can test that is going to help me understand how this is manifesting, how it's expressing in the body? And lastly, is there an intervention? Is there something I can do? If these clinical questions can't be answered, then you're buying data, not clinical value. Okay, so now I'm going to jump forward again. I am going to show you in a demo soon what we call the interactive blueprint report. It is built only for clinicians, and it really, the dream of the company amongst many things was, how do we make the report so easy for you that you can find the answers you're looking for in 30 minutes or less? I hear these terrible stories about clinicians who are spending hours going through SNPs and trying to figure out what to do, or going through multiple panels, a diet one, a brain one, a sport one, a health one, and trying to correlate across them. We never want you to do that. So I'm going to show you this interactive blueprint report and how we've achieved that. The other thing to mention is that when we started out with Rupa members could only access the PDF version, which is the patient-facing version. And so it's been super exciting. I think it's been like 2-3 weeks now where we've done a technical integration with Rupa, with their amazing team and my amazing team, to be able to offer this interactive version. And I'm going to do a demo in a few minutes and show you why this was such a game changer for clinicians working with genetics. In addition, we have this incredible community platform. We have about 6,000 clinicians there. It has education, it has continuous education, it has mentorship. You can ask questions, it has live events, and it's open and it's free. We have a free level and we have a $99 a year level where you get lots of additional stuff. If you go into the platform, it's just 3x for community. you'll be able to find it, and it'll tell you what is the difference between the elite access and what you can get. But just to know that this is a platform where you can feel safe. You can feel nurtured, no matter whether you're new to genetics, you've been dabbling in genetics, or you really want to own the space. The 3x4 community is available, and it's open to any clinician anywhere in the world, irrespective of what genetic test you're using. It's a place to upskill. Okay. And lastly, and my last slide before I jump into the demo, I think our timing is perfect, is mentorship, right? Mentorship is key to what we do. We know that when you're sitting with labs, when you're sitting, especially the functional integrated patients, they're complicated. They bring a lot. We're trying to discover it. We realize that education is great. We love it. But you really want to be able to reach out to someone, book a session and say, am I on the right track? Am I looking at the right pathways? Will this SNP have a big impact? How do I correlate this biomarker with the SNP? So in that 3x4 community platform, you can book mentorship. We have a getting started mentorship, which is really helpful. Unlimited group mentorship, you can book as much as you want. And then we have one-on-one mentorship, which is available if you're that elite access that I mentioned. Okay, I'm now going to share. So I'm going in through the 3x4 portal to show you, but of course you'll be accessing it through Rupa. You don't need to go through the 3x4 platform. I just want to do it this way. Okay, so now what happens is when you go in to your Rupa platform, you will have the option of downloading the PDF, as you always could before, which is the patient-facing one. And you will also have the option of opening the blueprint. And what you're seeing in front of you now is what we call the interactive blueprint. So on the left-hand side are all those 36 biochemical and metabolic pathways we spoke about. And on the right-hand side are all our genetic results. And as you can see, everything's color-coded. Remember, dark purple, genes are having a very high impact. Purple are high impact, blue is medium. And it actually says they're very high, so you can always be reminded. Okay, so 2 things about this platform that make it such a game changer. The first is you can filter it. So if you look at the pathway buttons, all these white buttons are filter buttons. So you can say, I'm only interested in cardiovascular, and it'll bring up all the cardiovascular pathways. All the other pathways are not shown, and give you the color. All right, you can say, I'm interested in a very high, dark purple, very high impact, and I'm only interested in weight. I want to see what's happening with my patient. This sample report, this patient has a lot of stuff going on in their weight loss journey and how they're experiencing calories and eating behavior and appetite and hunger and satiety. A lot going on with this patient. Okay, and you can do the same thing on the gene side. You can say, I only want to see very high, I only want to see cardiovascular, but I want to see the genes that are showing up. All right. The second thing about this platform is this clinical guide. Now, we call it the clinical guide, but what it is an interactive nutrigenomics textbook. So many years ago, every 2 years, I would publish in a book what I called the SNP journal, and basically it would be all the SNPs that were of interest, and there would be a page. This is it, this is what it does, etc. And about 2 weeks after every time I published it, I wanted to add more or edit something or update something. So it was a horrible idea to keep publishing. So instead, we moved to building an interactive version of it. And in fact, we expanded it so it didn't just have information on genes, it had the clinical recommendations as well. And I'm going to show you what I mean. This clinical guide is embedded into this report. Now, if you work directly with 3x4, you need to have that elite access at $99 a year to access the clinical guide. But as part of our partnership of working with Rupa, we are giving you the clinical guide for free, and it is embedded in the report. There are still many wonderful things about elite access, our presentations, our slide decks, one-on-one mentorship, masterclass series, but essentially you're getting the clinical guide, and I'm going to show you how amazing it is. Okay, so now I'm going to do a demo to show you how I use this and why it's so valuable. So I'm going to unclick. We're going to start from 0. When I see a patient, I always want to think big picture, where are the genes having the greatest impact? So one of my colleagues, Nikki Grant, who's one of our mentorship teams, brilliant, used such a great analogy. She said, why do we start at the pathway? So the pathway is starting think of a drone flying over a forest, getting a picture of what's happening in the forest. How big is it? How tall is it? The color of the trees, the color of plants. When you land the drone and you're in the trees, you're then at the SNP level. So up above, we're looking at the pathways, big picture, where the genes having impact. Then we can land the drone and say, okay, what is happening with this tree? Why is it not growing well? Why has it got rot? What are things? So that's the same way we approach our 3x4 report is I always start at the pathway side. I want to think about where the impact is, and then I'll go to the gene side. So that's what I'm going to do. So traditionally, the way I would do it is I would either click on very high and high. I'm going to start off just by clicking on very high, and it's going to show me all the pathways that are dark purple. You can see there's quite a lot of them. This is a sample report, and we've chosen a report that has a lot of purple, and not every report will have as much purple. Okay, now when I see a patient, I always go upstream. I always want to think, are there cellular pathways that are being impacted by the genes? Because if I don't resolve these core pathways, it's very unlikely I'm going to be effective downstream. And you can see in this sample report, we've actually got 3 cellular pathways that are dark purple: inflammation, methylation, oxidative stress. That needs some work. So for me, for the first probably 3 to 4 weeks of working with this patient, this is where I would focus. So let's explore this some more. I'm going to pick one of these, and I'm going to go deeper into it. I could judge within them. I'm going to choose oxidative stress. So when I choose oxidative stress, you can see all the other pathways are grayed out. Now, when I click on anything on the left, it's going to filter the right. And when I click on the right, it's going to filter the left. So when I clicked on oxidative stress, what happened on the right-hand side? Before, we had all the genetic results. Now you can see there's only some genetic results and all the other genes have been grayed out. Why? What you're seeing on the right-hand side is the polygenic group for oxidative stress. So all the genes that are showing contribute to oxidative stress in some way. Remember, pathway-based analysis is a polygenic score for a biochemical and metabolic process. So you're seeing that group of genes. There they are. Okay, now, so in addition, in case you didn't notice, the color is also there. So I can immediately see which genes are impacting oxidative stress, but I can also see which genes are having the greatest impact. And you can see the dark purple, MSOD and PPRGC1A are having the greatest impact on this pathway, and then ENOS and GSTM1. Okay, this is what we call the interactive blueprint, and you can play around and you can click. But now we're going to go into the clinical guide. So to get into the clinical guide, remember, clinical guide, interactive textbook. We click on these little half triangles here. So I'm going to click on the triangle. Now, the way we built this clinical guide is it's built in layers of depth. So, kind of a little bit superficial, deeper, deeper, deeper. And because it's Rupa, you have access to all the levels. Okay. I am going to, this is the top level. I don't really use this so much because it's kind of hard to see the screen, but essentially what it's giving you is 3 diet, 3 supplement, and 3 lifestyle recommendations. Remember, for every pathway, 36 of them, for very high, and medium, you are going to get 3 diet recommendations, 3 lifestyle recommendations, and 3 supplement recommendations. I'm going to show you a level down, and this is the level I usually work at. The first thing, we're in the oxidative stress page, is you're getting, in purple here, all the genes that form that polygenic score. Wherever anything appears in purple, you can click on it and it'll take you through to a page. So if I click on GSTP, you can see my cursor, it gets a line underneath. I can click on it and it goes through to a page about the gene. I can learn all about the gene. As I take you through this, the one thing you'll notice is what we've tried to do is create a platform where you never have to do the work that we should do for you. You don't have to go to PubMed and do a search. Tell me about this gene. What does it do? Download some articles. Does it change an enzyme? Is it up? Is it down? What should I do about that? All right. Everything lives in this platform. Okay. This is actually showing you how the picture changes. So we didn't go through it, but in our PDF report, we have these beautiful infographics that talk about the pathways, and the genetic result changes, the picture changes. So every patient's report is completely different because the image evolves depending on the genetic result. Okay. Now we go down to the recommendations. So remember, every pathway, diet, lifestyle, supplement. We've got 3 diet here, 3 and 3 supplements. Everyone in our clinical team is functional and integrative trained. So you'll see kind of a correlation with the type of things you would be learning through any of those platforms. Okay. Every single recommendation has a question mark. That is the why. Why did our clinical team think this was a useful recommendation for you to use with your patient if we want to address oxidative stress? So these, you can click on them all and they'll tell you our why. So we'll look at an example now. If you look at diet and you look at number 2 in diet, eat foods rich in zinc, copper, selenium, and manganese, such as seeds, nuts, leafy greens, and legumes. Question mark. Why is that a good idea? Because foods that supply cofactors for antioxidant enzymes. Great. Now I know why that would be a good idea. Remember, anything in purple you can click on. I click on zinc, copper, selenium, manganese, and I get a food list. All the foods that are highest in manganese and what would be an appropriate serving size. All right, let's jump back. We'll close it up. Okay. Same thing for lifestyle. We've got all the whys, and then we go down to supplements. So we are brand agnostic in supplements. We don't sell supplements. We don't have a company that sells supplements. We don't pretend and then actually sell supplements. So you only get the active ingredient from us. We don't recommend brands. If you reach out to us, we have through mentorship, we're happy to share, but we don't do that. So we give the active ingredient and we give a recommended dosage. You are the clinician. You are knowing your patient. This is not telling you what to do. This is just a resource. You can use as much of it or as little of it. And also just to note, if I do that, I can copy and paste everything in the clinical guide. I can take these recommendations, put them into a Word document, brand them with your logo, and share them with your patient. Okay. In addition to diet, lifestyle, and supplements, we have done what we call functional testing or test pairing. And this is why we love our partnership with Food for Health. We know genetics gives insight. A test gives the status of what's happening. So for every genetic pathway, we say what would be biomarkers that relate to that pathway. So you can see this is a blood test and some of the biomarkers that would relate to oxidative stress. And of course, why? Why would copper be insightful? Well, it's a cofactor for the gene, a very important gene. We're going to look at that just now. So we want to make sure that we've got the cofactors for the gene. Okay. In addition to blood, you're going to see organic acids. We always include organic acids. Which organic acids relate to oxidative stress and why? We've got hormone tests. So every single pathway will have different tests listed. All right, let's jump back. So we went in on the pathway side, remember, drone above the trees, and we looked at what the genes having an impact, what kind of diet, lifestyle, supplement recommendations could we use for that, and are the other tests we should be doing. Now we're going to go to the gene side, and this is really a helpful place that you never have to go and search for information about the genes. So I'm going to click on very high. Say, show me the pathway, show me the genes that are having the greatest impact. And you can see it starts off as the dark purple SNPs, then the purple, blue, and then it'll go down to the yellow SNPs. And then it'll go down to these, which have no impact on this patient. Okay. I can click on any of these genes. So I'm going to click on MSOD, manganese superoxide dismutase. It's a very, very important enzyme inside the mitochondria that is part of the process of kind of quenching superoxides. So we give you the name of the gene, the name of the SNP, the genetic result for your patient, and then the impact. Now you can see here, remember we do everything in colors. There's actually 3 colors. When you see multiple colors next to a SNP, it means that there are enzymes that have multiple impacts in biochemical processes and not only a single biochemical process. Okay. And when we look to the left, remember, click on the left, just click, it filters the right, click on the right, it filters the left. We look to the left and it's telling me what pathways this manganese superoxide dismutase enzyme is actually impacting, and to a degree how much. You can see it's color coded. Okay, now we're jumping into the clinical guide. So we jump into the clinical guide and it's 2 levels for the gene side. The first is a SNP summary. I use these all the time. I don't remember every single SNP that we use. I know some of them better than others. Sometimes I just want to go remind myself, what does this gene do? What does this SNP do? So you're getting a summary, and the summary has like a formula for you. The formula will always be, what is the enzyme the gene codes for, or the hormone, or the neurotransmitter? What does that enzyme do? What is the impact allele? So which of those nucleotides are having the impact? In this case, it's the T. And what does that T do to enzyme activity? Well, in this case, it decreases about 30-40%. And then the last thing we'll include is, but what is the result or the impact of having the 30 to 40% less enzyme activity? So that's all in one little paragraph, like 2 sentences. And of course, you can go deeper. So now we're in the MSOD page. The first paragraph is the biochemical context of that gene, of the SNP, what's happening, what does it do, what's going on. We'll tell you which other pathways it appears in, the impact allele, we give the RS number. Very important. What is the impact of the T allele? What is the impact of the C allele? Sometimes one will be protective and one will change enzyme activity. And then lastly, we have something called clinical notes. So I always describe clinical notes as the so what, why should we care notes. It's kind of a summary of cofactors and diet and lifestyle If there are other genes we should be thinking about, we'll mention them in the clinical notes. In this case, you always want to think about GPX and catalase and see if there's SNPs in there as well, because then you really know there's a lot of intervention points in oxidative stress you need to address. And lastly, if there are tests, biomarkers that relate directly not to the oxidative stress pathway, but to the SNP, it'll also be mentioned here. And if you click on that, you can actually learn more about the 8-hydroxy, all the pathways that it influences. We could go in here and it'll tell you what is the relevance of the biomarker, what is the optimal range, and what are the SNPs that relate to it.
Welcome to Rupa Health, the best place to order, manage, and track results from over 30 different lab companies in one single place for free. It's going to take you under 2 minutes to sign up, and you can order any functional medicine lab for your client in under 30 seconds. Let me show you how it's done. So here's our beautiful interface. I'm typing in the name of my client, selecting the lab that I want to order for them, and hitting send. From there, Rupa and their amazing team handle the rest. They email the client, collect payment, and even offer an interest-free 3-month payment plan. We've also built the world's largest library of information about chronic health conditions, the lab tests that can help you find the root cause, and the evidence-based interventions that you can use to help people heal from them. It's called the Rupa Health Magazine. There, we have in-depth articles about almost any health condition you can imagine. And we give you step-by-step protocols that other clinicians have used to help their clients heal and that are verified by evidence-based sources. You should totally check it out, and it'll transform your practice. And we can't wait to see you. So make sure you sign up today at rupahealth.com.